everyone, it's 11 o'clock, so let's get started. Thank you all for joining us for our webinar today. I'm Jackie Carville, the Marketing Director here at Immunochemistry Technologies, and I will be moderating this webinar. You may have noticed that your phone has been muted. However, we do encourage you to ask questions along the way. Simply type your question into the chat dialog at the right and select Send to Host. If you have any technical difficulties during the webinar, please send your chat to me and I will do my best to get you up and running. We will be posting the video recording of this webinar later this afternoon, so look out for an email afterwards with a link to the recording. And finally, look out for some special information from our speaker. So I'd like to introduce our speaker today, Dr. Michael Olin. Mike is an assistant professor in the Department of Pediatrics at the University of Minnesota. Today he will be presenting how central nervous system tumor-derived exosomes suppress an anti-tumor response. And with that, I'm going to go ahead and turn it over to Mike.
Well, thank you very much for that introduction. As I tend to talk fast, as my wife used to tell me, please feel free to slow me down. If there's any questions, please feel free to ask. I do have a lot of data I'd like to talk about. So if we are running short of time and I don't answer right away, I will get back to your questions. So I work at the University of Minnesota, sunny Minnesota. It's lovely a couple months out of the year. And I focus mostly on brain tumors, neuroimmunology, oncology, and how we can take our bedside science to the clinic. And ever since I was a postdoc, I've been kind of focusing on immunosuppression through opioids and tumors. And to really extend my research, I fell across this company, ICT, with very novel and very strong, powerful immunological assays, especially as it relates to Cas-based activity. A lot of this was published in prior manuscripts, if anyone wants to look it up. But as I said, I work on immunosuppression and what we can do to better improve our immunotherapy for our tumors. Now, I didn't want to do that. Okay. So a while ago, it was reported, mostly in the peripheral system, that the draining lymph nodes, those lymph nodes closest to the tumors, are immunologically suppressed. Now, this is in part due to metastasis of tumors into the lymph nodes, but this group showed that the closer to the tumor site, the less capable you are of inducing both of a mitogenic as well as an antigenic immune response. So I asked the question, what about brain tumors? Now, brain tumors do metastasize somewhat into what's called the cervical lymph nodes, but it's relatively rare. They usually metastasize within the central nervous system. So what I called, being that the CNS doesn't have a traditional lymphatic system, the cervical lymph nodes I'm going to refer as the draining lymph nodes. And that's because the CSF filters antigens through the brain into the deep cervical lymph nodes, and then it's reabsorbed. So it acts as a draining lymph node. So what we did is ask a simple question. If we go close to the tumor site, in this case the cervical lymph node, are we inhibiting the ability to mount an anti-tumoral response to our immunotherapy as it relates to brain tumors? So to do that, we gave these little mice mycegliomas called GL261s. They're well-defined tumors of the central nervous system in rodents, in mice specifically, and we vaccinated with ova plus an antigen or adjuvant, in the back of the neck for drainage into the cervical lymph nodes, in the foreleg for drainage into the axillary lymph nodes, and the hind leg to get further away from the tumor and drainage into the inguinal lymph nodes. Now, it was really surprising that it showed that if we went into the back of the neck, we are incapable of eliciting an immune response, at least a potent immune response. And this was true also for the axillary lymph nodes or the hind forelegs. However, if we went back into the hind leg, further away or far from the draining lymph nodes, the cervical lymph nodes, or the tumor site, we are capable of eliciting an immune response, both shown through a syncytial or ova-specific T cell proliferation, as well as a functional response. And going back, that black bar just indicates that non-tumor bearing mice were able to elicit an immune response, showing that this was not just a function of the cervical lymph nodes itself. So as I said, we were able, if I press the right button here, able to elicit a functional immune response, which actually translated to an increase in survival. So we asked a simple question, well, not necessarily so simple, but the question, what is suppressing the ability to induce an immune response to our immunotherapy? So what we did is we took just wild-type splenocytes, pulsed them with ova, and then we took what's called an OT1, a transgenic T cell from OT1 mice that already has the TCR. Can you hear me now? So it already has the TCR TCR ready for the syncytial peptide. So you can see here that if we use this system, pulse the splenocytes, add the T cells, we are able to elicit a T cell response measurable by interferon gamma. So we took the CSF from tumor-bearing and non-tumor-bearing mice, and here you can see from non-tumor-bearing mice, we really had no functional differences. Now, the CSF we took, we actually looked at, made sure that it was clean. We devoided them of all common proteins, and we made sure there were no suppressive cells in there. We even freeze-thawed. So we thought if there was a difference, it's other than a myeloid-derived suppressor cell, T regulatory cell, or another suppressive cell. And what we saw was that if we use the CSF from tumor-bearing mice, we were incapable of eliciting an immune response. So now we know that this is something other than a suppressor cell. Something's really suppressing ability, specifically or hopefully coming from the tumor site, hypothesized coming from the tumor site, that we are inhibiting the ability to mount an immune response. Well, one of those possibilities are what's called exosomes. Now, exosomes are little blips of cells. All cells give off these exosomes, and they carry a genetic RNA, DNA, they carry proteins, which recapitulate that of the parent cell to a certain extent, especially when it comes from surface proteins. Now, as a little disclaimer, exosomes, by definition, are less than 1,000 nanometers. So we're showing here we have a heterogeneous solution of exosomes and microvesicles. They're virtually the same thing. However, central dogma says less than 100 nanometers is an exosome, larger is a microsome. For the purpose of this talk, I'm just going to call everything an exosome. So we're showing here that we have, a nice heterogeneous population of exosomes and microvesicles. They are GL261 or tumor-derived exosomes, which are luciferin positive. So we show that we're still carrying that activity and that they are positive for typical exosome markers CD63, CD9. So we're relatively convinced that we have some purified exosomes. So we went with the hypothesis that tumor-derived exosomes will inhibit the ability to activate an immune response. To test this hypothesis, what we did was we took non-tumor-bearing animals, okay, these did not have tumors, and we vaccinated them prime boost experiment with ova or ova plus exosomes. And what we saw off our hypothesis that we're going to suppress the ability to actually induce an immune response was actually quite the opposite. We actually enhanced a proliferative response, looking again at the syncytial CD8 positive TCR populations. Moreover, we showed that we were incapable of really altering a functional response. Now, this is all in vivo. Recently, by Grainer et al., showed this in vitro that that adding certain concentration of exosomes enhanced proliferation, but didn't change a functional response. So we went on and showed that that they were incapable, in this case, of eliciting a tumor cyto response, but we really didn't see much difference in functional responses as it comes to cytokines. Well, we started thinking that you really don't go to the doctor and say, hey, we're going to get a brain tumor next week, okay? You go to the doctor because you're showing clinical signs. So we went back to the drawing board, and we pre-treated our animals with exosomes, purified exosomes from GL261 tumors on day 0, 1, 2, and then we continued the exosome treatments as we gave the vaccinations with either OVA plus poly-IC plus exosomes. So this time what we saw was that when we gave exosomes plus OVA, as I just told you, we actually enhanced the proliferative response. However, if we pre-treated the exosomes and continued on, we saw a significant reduction or suppression in an immune response. So, as Dr. Grainer said, that they showed that it's most likely a concentration issue. We further went on and showed that if you pre-treat the animals and then vaccinated them, you significantly suppress the ability to induce both a cytokine response and, again, we are incapable of eliciting a cytolytic response. So the CTL responses are really a great measure of immunogenicity, and we're going back now with the apoptotic probes that ICT makes in a total cytotoxicity kit. We can actually differentiate apoptosis from necrosis within our sample, so we could focus on the actual mechanism of killing by flow cytometry, and we're in the process of doing that right now. So we show that our hypothesis is potentially correct, that tumor-derived exosomes suppress the immune response, or the ability to mount an immune response. This is true for tumor-derived exosomes, however, not non-tumor-derived exosomes. So we use DC cell line, dendritic cell line for mirroring dendritic cells, and we collected the exosomes and repeated all our experiments, and we see that non-tumor-derived exosomes fail to suppress an immune response. So this is specific for the tumor. So we decided to go downstream a little bit and look within the lymph nodes themselves. Now, the data I'm going to show you in a few minutes, I've done on both the inguinal lymph nodes and the cervical lymph nodes, and we got the exact same responses. So this is not, again, selective of a specific draining lymph node. What we did is went and repeated the previous experiments where we gave exosomes plus OVA and adjuvant, or OVA plus adjuvant alone. Now, the OVA significantly, I don't have the saline control here, the OVA significantly enhanced or expanded the draining lymph nodes. And like I said, it doesn't matter if these are cervical or inguinal. But what was really interesting was any of the treatment groups, rather we pre-treated them or not, didn't pre-treat them with exosomes, you saw these well-defined germinal centers with these thick T-cell margins. But what was really interesting is if you look within the germinal centers themselves, you can see these hyperactivated B cells. Now, this was only found within mice that received exosomes, and here's even a more close-up where you can see them. The B cells are becoming hyperactivated, and they're all tunnel positive, so they're dying. Something's killing them. And again, this is true with tumor-derived exosomes only, because if you look in the non-tumor-derived animals, you see some expansion of germinal centers, but you don't really see any of these hyperactivated B cells. So we went back to our mouse model, and we decided to, instead of giving the mice tumors, we actually gave them purified tumor-derived exosomes. So we did this using an LSAT pump and for continuous flow of exosomes. And what we showed is that we can recapitulate that of the exosomes being thrown in right on the lymph nodes, that these are actually coming from the tumor site. They're in the exact same spot as our tumors, and they're flowing through into the draining lymph nodes, inducing tunnel-activated, hyperactivated B cells. And this manuscript, by the way, is being submitted within the next week or so, so hopefully it'll become available within the next couple months. So we're showing that it is tumor-derived exosomes are flowing through, presumably through the CSF, into the draining lymph nodes and inducing a hyperactivation of the B cells, which induce tunnel staining or tunnel positive B cells, or killing the B cells virtually. So we decided to look at more mechanistic experiments. Now, it was reported recently that activations of toll-like receptor 4 in tumors themselves induce the synthesis of various soluble factors, and that blocking the toll-like receptor 4 actually enhanced immune responses or reversed the tumor-mediated suppression. So we went ahead and used the HEC293 cells that are commercially available that are transgenic for toll-like receptor 2 and 4. And we saw that tumor-derived exosomes, not non-tumor-derived exosomes, induced an immune response or activated NF-kappa B, which is a reporter for toll-like receptor activation. So TLR2, TLR4 are activated by tumor-derived exosomes only, not non-tumor-derived exosomes. And this is the same with human tumor-derived exosomes. We took glioblastoma multiforme cells that we have, and we purified the exosomes, and we showed that the toll-like receptor 2 and 4 are becoming activated using a human monocyte cell line to derive non-tumor-derived exosomes, show that this is true only with tumor-derived exosomes. But interestingly, being that exosomes and microvesicles are chock full of RNA and DNA, we failed to see activation of toll-like receptor 7 and toll-like receptor 8. So it looks like they're running through toll-like receptor 2 and toll-like receptor 4 from tumor-bearing exosomes, which I guess with doing proteomics wasn't overly surprising given the significant difference of heat shock proteins on or by tumor-derived exosomes, not non-tumor-derived exosomes. So are there any questions so far?
No questions so far, Mike, but everyone feel free to chat in those questions to me, and I can field them to Mike throughout the webinar.
All right, thank you. So we went one step further and tested our hypothesis that these are toll-like receptor-mediated immune response, and took toll-like receptor 2 and toll-like receptor 4 knockout mice and treated them with our exosomes. And here we showed that our exosomes suppressed a wild-type mouse, suppressed the ability to mount an immune response, which I've showed you before. The same was true with toll-like receptor 2, but not toll-like receptor 4, indicating that this is potentially a mechanism through toll-like receptor 4. So we went in and actually looked at the lymph nodes again to validate this, and hopefully you can see this. It's a little small, I apologize, that we do see the hyperactivated B cells, tunnel-positive B cells, in toll-like receptor 2 knockout mice, but we do not see them in toll-like receptor 4 knockout mice. So this further indicated that tumor-derived exosomes, not non-tumor-derived exosomes, have the ability to suppress an immune response, potentially through toll-like receptor 4, inducing tunnel-positive B cell hyperactivation. Now, I didn't show this, but we do have evidence that the T cells surrounding the hyperactivated B cells are caspase positive. I didn't show you that because we're repeating it, but we showed that there is caspase activation, presumably on T cells, and we're kind of hypothesizing that these are vesicular T cells, because vesicular T cells need B cell activation to function. So we decided to take this one step further and look at what's called inflammasomes. Now, these inflammasomes are activated and they produce IL-18 and IL-1 beta, and these cytokines are really important for the differentiation or the advancement from innate to adaptive immune response. So we decided, although there's several different pathways, to look at the role of inflammasome and tumor-derived exosome-mediated immune suppression. So we went to our human experiments or human cell lines, and we're showing here that on normal monocytes, exosomes will elicit the secretion of IL-1 beta, or they'll make IL-1 beta cytokine. However, if you knock out the inflammasomes, you do not get this IL-1 beta activation. So now we're showing that potentially, again, through toll-like receptor 4, these are inflammasome-mediated mechanism, incapable or inhibiting the production of IL-1 beta. So we went and got some caspase-1 knockout mice. Now, caspase-1 is downstream of the proteins in inflammasomes that are required for active inflammasomes. So we went in vivo, and we actually showed that this is true. If you use caspase-1 knockout mice, you don't get the hyperactivation that you normally see in wild-type mice. So this brought us to the summary, and basically what's being submitted soon is that tumor-derived, not non-tumor-derived exosomes induce immunosuppression. Tumor-derived exosomes induce immunosuppression through potentially the toll-like receptor 4 pathway, which lead to inflammasomes, and this leads to the hyperactivation of B cells, which lead to the tunnel positive or killing of the B cells, and potentially the knocking out of follicular T cells. So that's what I wanted to talk about, tumor-bearing exosomes and their role in immunosuppression as a potential way that the tumor can evade the immune system. And I'd like to acknowledge people in my laboratory, collaborators, as well as other supporters, the Randy Shaver Cancer Foundation, Masonic Cancer Center, and Immunochemistry for actually supporting some of this with their reagents. Thank you very much. I'll take any questions you have.
Great. Thanks a lot, Mike. Again, feel free to chat in any questions if you have them. Otherwise, if you'd like to contact me directly at jackie.carville@immunochemistry.com. We can follow up with you personally after the webinar as well. I'll also be sending this webinar recording out within the next day or so. For those of you interested, our next webinar will be held on January 27th, and we'll answer the question, how healthy are my cells? And here you'll learn about new tools to assess overall cell culture health, as well as methods to evaluate experimental treatments on those cells. And you can register for that particular webinar on our webinars page. If you can think of any questions after the webinar again or have ideas for future webinars, you can also fill out the Contact Us form on our website or reach us through social media on Facebook, LinkedIn, or Twitter. So thanks again for that excellent talk, Mike, and thanks everyone for joining us. Have a great day.